Hey guys, welcome to my channel Tuli's Biology, and today again I have come up with a new topic. The topic name is Continuity of Life but part 5. This video is for Madhyamik students, these are Madhyamik students, Continuity of Life part 5. So I'll start with my video, but before that please subscribe to my channel if you haven't subscribed it. Like the videos, share the videos, watch the videos, and obviously make sure to follow me on my Facebook page, my mail ID as well as Instagram page, and all the links I have provided in my description box. And if you need any notes or any diagram or anything related to this chapter, then do let me know in my comment section. Also you can let me know in my Facebook page or my Instagram page. I will obviously follow up you guys, and I will obviously provided all the things that you will need. So let's just start with the continuity of life part 5. In my previous video I have done the mitosis part. So in this video I will just quickly recapitulate that one. Mitosis, as I have told you, that's the equational division. So before dividing, they just double up their content. Okay, so mitosis cell division, it is consisting of 4 stages: prophase, metaphase, anaphase and telophase. So what happens in the prophase? In the prophase, that is the first phase of the mitosis. Here the chromatin network, they just lose water, and instead of thin and long, they become short and thick. And nuclear membrane, nucleolus, they just disappear. Coiling of the chromatin network takes place. So these are the characteristic features of the prophase. In the metaphase, what happens? Meta, the word meta comes from middle. So here the chromosome starts to it is getting arranged at the equatorial plane or equatorial region. See, and also another thing is that the centrioles move to the poles, starts to move to the poles in the prophase only, and they starts to form the aster rays in the prophase only. So the beginning of the formation of the spindle fiber starts in the prophase only. Okay, but in the metaphase, what happened? The chromosomes are arranged at the equatorial region, and now the spindle fibers are attached with this aster rays. Okay, so this is the metaphase. Here this characteristic feature is being done. Next is the anaphase. Here anaphase means the chromosomes will move towards 2 opposite poles due to the shortening of this spindle fiber. Okay, due to the shortening of the spindle fiber, the chromosomes will tend to move towards the poles. So this is known as anaphase movement. So this is being done in the anaphase. Now last one is the telophase. Here totally the chromosomes has been moved to the extreme poles. The despiralization or uncoiling of the chromatin network has taken place. That means from chromosomes now it is being converted to chromatin network, and reappearance of nucleolus and nuclear membrane has also taken place in the telophase. So this is the mitosis part. Now after this we will jump to the next part, that is the cytokinesis, which is the division of the cytoplasm. Now in case of plant cell, what happens? There is the organelle named Golgi body, which will pour out its secretion, and as a result of which it will create a thin plate at the middle of the cell, and this plate will elongate. This is called phragmoplast. So it will just go deepens, and it will just get bigger and bigger, and it will form cell plate. ঠিক আছে, যেটা ফ্র্যাগমোপ্লাস্ট বলছিলাম ওটাই এখন বড় হয়ে সেল প্লেট হলো, and now what will happen? This cell plate will ultimately divide the one mother cell into 2 daughter cells. This happens in case of plant cell, but what happens in case of animal cell? Here animal cell invagination or furrowing takes place. That is, a small groove has taken place at the middle of the cell, and this groove now only it will deepen, and ultimately it will give rise to two daughter cells. So in this way the cytokinesis takes place. Now we will move on to the next topic, that is the cell cycle. Sorry, not cell cycle, sorry, extremely sorry, the significance of mitosis. As we all know that what are the significance, that is the importance of mitosis, that is obviously it helps in the total increase of the cell number, total growth, total repair, growth and development, any kind of repairing or regeneration or healing the wound. All these things are being done or are being controlled by the mitotic cell division. So here the topic of mitosis is done. Next we will go to the topic of meiosis. So this is the meiosis topic. In this topic we will understand what is meiosis and what is the significance of it, how it is done. So meiosis is a type of cell division that takes place in our gamete cells. Okay, that is, it leads to the formation of sperm and ovum. So what happens over here, the meiosis is termed as reductional division. That is, the mother cell is having 46 chromosomes, whereas the daughter cells will have 23 chromosomes each. But why it is done, how it is done, that we need to know. And as the content of the mother cell gets reduced to half in the daughter cells, that's why it is called reductional division. So how it is done, that we will. First of all, meiosis takes place in two steps. One is meiosis 1, another one is the meiosis 2. Meiosis 1 is just like the mitosis only. That means what happens in this case, we will see. One thing that you have to keep it in mind, that meiosis takes place in the gamete cells only, that is in the sex cells, not in the body cells. Mitosis takes place in the body cells, meiosis always takes place in the gamete cells. Okay, so first of all 46 number of chromosomes done. Now this will double up its contents, that is it will become 92. So when it is getting double up its content, so it will give rise to form two daughter cells, each will contain 46 chromosome, 92 getting divided to two daughter cells, 46 chromosome. So this is our meiosis 1. Now from each of these 46 chromosome, again two daughter cells will come out, but here no content will be double up. Here the 46 chromosome or 46 content will be divided into two halves, that is here 23, again here is 23. So this is our meiosis 2. Actually this reduction takes place in this meiosis 2 phase, that's why it is called reductional division. Okay, it is taking place in the gamete cells or germ cells, resulting in the formation of the gametes. Clear? Now we will see the important characteristics of the meiosis. First important characteristic is that separation of the homologous chromosome. Now what is homologous chromosome? We know that the chromosome or the genes always they exist in pairs. Now the pair of gene which are structurally as well as functionally similar to each other, exact similar to each other, those are termed as homologous chromosome, like this two. Okay, now this homologous chromosome is present in both sperm and ovum. So as I have told you the 23, so here two sets cannot present, only one set. So here separation of the homologous chromosome first is taking place. Obviously 46 means what? 23 homologous pairs are present. Okay, so here 23 means what? That is 46 chromosome is not present. 46 পেয়ারটা মানে হচ্ছে আমার কাছে 23 খানা হোমোলোগাস পেয়ার রয়েছে, কিন্তু আমি যখন গ্যামেটটা তৈরি করছি তখন তো আমার কাছে 46 খানা ক্রোমোজোম নেই, আমার তখন আছে 23 খানা ক্রোমোজোম। তো ওখানে হচ্ছে ফার্স্ট আমার সেপারেশন অফ দ্য হোমোলোগাস ক্রোমোজোম টেক্স প্লেস ইন দিস ফেজ। ওকে, দ্যাট মিন্স হোমোলোগাস পেয়ার দুটো থেকে এবারে আমি একটা পাচ্ছি, যেটা একটা এই গ্যামেটে আসছে আর একটা এই গ্যামেটে আসছে, মানে এই দুটো হোমোলোগাসের মধ্যে থেকে একটা এখানে আসলো আর একটা এখানে আসলো। So this is the separation of the homologous chromosome taking place. This is one of the characteristic. Second characteristic is that reduction in the chromosome number. Obviously, as you can see, from 46 it has become 23. Separate হচ্ছে, ভাগ হচ্ছে, তো obviously it will become 23. Next is the crossing over. Now what is crossing over? Now this homologous pair, one of the homologous chromosome of father, it will combine with the homologous chromosome of the mother. So what will happen? They will just exchange a segment. So as a result of which we will get So this is the exchange of the segments has taken place. This phenomena is called crossing over, and during this exchange this X-like structure has been formed, which is termed as chiasma. So this crossing over always takes place in the non-sister chromatids. Non-sister chromatids of the homologous pair. See, non-sister, দুটোর মিল নেই। সিস্টার ক্রোমাটিড কাকে আমরা বলবো? যে দ্য ক্রোমাটিডস উইচ আর কানেক্টেড বাই আ সেম সেন্ট্রোমিয়ার, দিস আর দ্য সিস্টার ক্রোমাটিডস, but these two or these two, these are the non-sister chromatids. So these non-sister chromatids will pair up with each other and it will form, or it will give rise to the chiasma formation, which will ultimately leads to the crossing over, that is the exchange of the genetical segments. Next is that what are the significance of the meiosis? First significance, it maintains the chromosome number. See, from 46 we are getting 23 chromosome, that is 23 from the sperm and ovum. So 23 will give us 46. যদি 23 জায়গায় আমরা 46 পেতাম, মানে 46 স্পার্মে রয়েছে, 46 ওভামে রয়েছে, তাহলে আমাদের নেক্সট অর্গানিজম উইল হ্যাভ ক্রোমোসোমাল কন্টেন্ট 92, যেটা কিনা পসিবল নয়. So that's why it maintains the chromosomal constancy. Second is that variation in the organism due to this crossing over. Third is that formation of gametes, that is formation of sperm and ovum takes place, or it occurs due to the meiosis process only. So this is all about the first part of the continuity of life. In the next video I will obviously go with the second part of the continuity of life. So make sure to follow my channel Tuli's Biology, and also make sure to follow me in my Facebook page and my Instagram page, which the links I have provided in the description box. So till then everyone, stay tuned to my channel. Bye bye and take care.